So I will introduce the next speaker now. Next speaker, Associate Professor Christopher Mansson, who has a research group at the Karolinska Institute. And he will be talking about examining the reliability and validity of neural variability in psychiatric neuroimaging.
Thank you. Awesome. Great. Thank you very much for the invitation and the chance to get to talk to you guys. Happy to be here. I'm a bit jet lagged after a conference, but I think I'm had a few cups of coffee, so I hope I can make it. So I'm a clinical psychologist as a training, so I think we're going to switch gears a little bit here from moving from cellular level to psychological level of understanding or the brain. I don't know if I want to use neuroscience in order to better understand the patients that I met in my clinical practice. And I don't know if I'm brave or maybe just stupid, probably both or a combination of both, trying to use neuroscientific methods to better understand our patients and find applications on how this can improve psychiatric treatment in the future. I know there's a long road ahead. I know that we are still trying to really understand the basics of how the brain is wired. I probably won't experience myself in my lifetime how this will become an application in psychiatry. But hopefully we're contributing to something that goes in the direction that I think is really important for psychiatry. Well, yeah, you probably know that mental health is a major concern. Not only the number of people who suffers from psychiatric illness and most commonly anxiety disorders and depression, but also the societal costs for this. But of course also the individual suffering that this is causing. And still today, what we are using as our primary method to understand psychiatric illness is verbal information. It's how we ask questions to our patients. We're trying to understand their suffering, and we then cluster patients according to their storytelling on how they experience their different symptoms. But even though this verbal information might be very important and useful for us, two very different experiences may also have a very similar neurobiological mechanism. So I think in order for us to really take the next steps in psychiatry, we need to understand the underlying biology. So we have treatments. Some of them are fairly effective, but still 40 to 50% of psychiatric patients do not respond to the current treatments that we have. And those are in best cases, the evidence-based treatments available. And that includes both pharmacological interventions like SSRIs, which are very common for anxiety and depression, but also psychological interventions like cognitive behavioral therapy, which is one approach that I've been trained in myself. Okay, so that's this is really important for me and why we are doing this job, and I hope we will be able to discover some biomarkers that may be useful in the future. The whole purpose of this slide is just how I started off my journey in using magnetic resonance imaging, both functional and structural MRI, in order to understand patients with social anxiety disorder. So social anxiety meaning, the type of symptoms that I'm myself experiencing right now, being in front of you, like feel like I'm shaking a little bit. My voice is a little bit. Those things would be absolutely horrible for someone with social anxiety, and they would either avoid it or they would experience this situation with extreme fear and anxiety. So I started off, and now my mouth also got very dry, so another symptom came up. So when you start talking about it, it actually becomes worse. Okay, so let's stop talking about that and talk about this. So my approach was to use fMRI and look at patients that we actually give treatment to understand what is actually treatment doing to the brain. And we used a pre and post design, so we follow patients we scan them before treatment and then after treatment. And this was fairly, this gave us some information. Oh, thank you. Wow. Wow. Another psychologist in the room. Wow, that was good. Okay, so, but actually the whole point of having this slide here is just to say, this is just too simple. If you really want to understand how treatment works and how people are affected, we need to sample much more data. We need to follow people more over time. Pre and post designs is not sufficient. Okay, so I'm going to trash my whole thesis here and saying that this was not enough. But another issue that I also experience within the whole field is test-retest reliability. So when we do actually test people at multiple locations with functional magnetic resonance imaging, the intra-class correlation coefficients are so low that we can really, I mean, I'm really questioning myself. Is this, how would we even end up having a biomarker if when we do the test, we will get different answers all the time? So these are two meta-analysis in the field, one to the left on functional connectivity and another paper to the right on task-based fMRI. And just to the average intra-class correlation coefficients here for functional connectivity is as low as 0.29, and for task-based is a bit higher, but still in the very lower range of what something to be a decent reliability. And for me, that's really something we should or I want to better understand and see if we can improve. A few other challenges within the whole neuroimaging field, not only in psychiatry, but of course that's in psychiatry as well, is the pre-post designs that I mentioned. And there's another, in our field, people are also saying that the reason why we have low reliability is that we don't acquire enough data. We need to scan people for longer times. So we typically have patients in a scanner for 1 hour, but there's people are saying that maybe we need to scan people for 2 or even 3 hours. But then again, is that really feasible? Is that even possible to have some of our most worst patients in a scanner for that extended period of time? I'm not so sure, actually. So I want to see. I think we need to go in the other direction to find short, feasible, and effective assessments. Of course, when we do our studies, we are not primarily interested in those actually individuals we include in our study. We're interested in generalizing this knowledge to new patients. So every time I get a new patient into my clinic, I want to make predictions for that person. And of course, this is a very important topic for us to find models that can generalize to new individuals. Reproducibility is, of course, also related to what I just said, but there is also in this domain of research suggestions of that to even make predictions at the individual level, we might need thousands or tens of thousands of individuals. And after a paper in Nature 2022, became, of course, very influential. And some people said that, oh, I'm leaving the field. I will give up. There's no chance we can contribute to this field. I usually pick up the supplementary figure number 17 in this paper. It's so nice that they hidden it so far away down in this paper. It's very nice because instead of having thousands of individuals in their own simulations, they actually found that if you do repeated testing and do the same scanning for more than one time per individual, you may end up having a decent sample size of even 30 or 40 people, which was a quite different story from the one that was told in the first main section of the paper. So, but okay, so my point is that we need to scan individuals more than once. And we need to take this time into consideration. So what actually happens over time, and that may give us important information. So I came across one of my postdocs adventures to Berlin at the Max Planck Institute for Human Development. Came across something that really caught my attention, because the whole way of thinking about that our brain actually dynamically change every second, and these oscillations or fluctuations in brain activity are actually meaningful. And this is what typically been named as moment-to-moment neural variability, and it's been very influential in studying the healthy brain, or and there's a whole literature on the healthy aging. And typically what's been found is that an adult, younger, healthy brain is more variable than an older, less accurate or less precise brain in making, like for instance, in cognition and memory. So having a brain that can variably respond seems to be a very good thing. And this whole idea of a brain that varies from moment to moment is not a new thing. I mean, it's been around for decades. And for those of you who work with frequency-based measures, EEG or MEG, you know that this has been more or less standard in the field. But for us who do bold fMRI, it remains to be an undervalued approach, especially in the clinical domain and especially in psychiatry. So to then be very, very clear about what we are doing, and what I plot here is just some typical bold responses of the occipital cortex, while I stimulate have individuals to watch visual stimuli on a screen. And as you can see, the bold response really fluctuates over time. But our models that we typically use in this field is that we average response over time. We say that during these time points during the experiment, we did certain things that we believe that these are the most. This is what we want to study, and these are the most important activations. But of course, by averaging, we also consider everything that varies as noise, and that's so instead of averaging, we actually find metrics that better can capture this variability. And so one simple way to do this is just to quantify this as the standard deviation. So meaning that each voxel across the whole brain can have its own deviation, or it would be quantified as the standard, each voxel standard deviation. So we really flip between between averaging and then instead looking at the standard deviation. Okay, so this now this was my short introduction and background to why I think it's important to study reliability, because it's been so far presented and been really low. I want to give you a few examples of how we can approach that, and also show you some examples of why this can become valid in psychiatric neuroimaging. So to give you an idea of just comparing the current what we call the mean bold approach, which is the average of the bold response to a certain time point, as compared to the SD bold for the same tasks. So when we compare mean bold and SD bold, we're talking about the same type of stimuli, same type of experiment, same recording, but we just quantify the signal in two different ways. And in this case, I have on the figure A and B, we have social anxiety disorder patients to the left and healthy controls to the right. And what these histograms shows is the distribution of all the voxels across the whole brain. So these are 50,000 voxels at 3 by 3 by 3 millimeters, so 27 cubic millimeters. As you can see in the purple and the green histograms, the SD bold test-retest reliability, quantified as the intra-class correlation coefficient, is up to 0.7 or even higher. And that's the average. Then, of course, you will see it on the right end of the tail that some voxels are, of course, much stronger. And when you look at the spatial pattern for how that activity is distributed, it's not only capturing what you would expect to be present for a visual stimuli, like in this case. So you would have the fusiform gyrus and the occipital lobe being the most reliable targets. But for mean bold, which is again our conventional method of how to look at the data, you would only capture reliable responses in the occipital lobe, whereas other responses across the cortex would disappear, especially if we set the threshold at an ICC at 0.5 here. Yes.
How is the retest with the mean bold? Because like for the standard deviation approach, you show like test-retest, one, test two. And for the mean, you
so it's two different tasks. So it's task one and task two.
Okay.
And
I thought it was two times the same one. That's what
no. So it's 2 time points in each of these histograms. Yeah.
The previous image, the 100 milliseconds potential, isn't it the visual evoked potential if it is in the occipital?
Mm-hmm.
The 100 milliseconds spike.
Oh, here.
Is it not structural, just structure based?
So when we sample bold signal, it's to a second, but here we have our repetition time for our bold acquisitions are usually 2 seconds, and for the data that I'm plotting here is actually a 2-second TR.
Oh, it's not milliseconds.
It's not milliseconds. No, this is actually seconds that we capture. Yeah. Mm-hmm. All right. Another thing I want to highlight here is when we compare our SD bold, we can do that in task, but we can also do that in typical resting state data. And if we trim our data going from 10-minute scanning to 5-minute scanning, and we do that we have in this case done 3 sessions per individual. And the take-home message from this figure is that if we keep the scanning time shorter, we will still have the same higher test-retest reliability for our variability measure, as compared to 3 other functional connectivity, REHO, and FALF, which are other commonly used resting state approaches in the field. So more data may not give us better data, actually may even worsen, but actually going in the other direction and having less data with a better metric may be the approach to go. Okay, there are so many things to say about this slide, but with a short time, I'm going to focus on a few things. So now we've seen that variability may be reliable, but is it biologically valid? I really wanted to test that idea with a classical twin design. So I approached a colleague of mine who's done an extraordinary job collecting almost 300 twins from the Swedish twin registry. And among these 300 twins, we have half of them are monozygotic and half of them are dizygotic. Then, of course, to test the heritability, then we would expect that monozygotic twins are much more similar than dizygotic twins. So using this very nice design and heritability estimation, we can estimate our brain's each voxel's heritability, and we do that with so this is the same data again from the same individuals, computed as either their neural variability or their mean bold response, and this is again emotional phases that we have the individuals to look at the screen. And we are very fond of using emotional phases as a stimuli in the psychiatric neuroimaging, because a lot of our patients also, most of our models on understanding the psychiatric illness and especially social anxiety disorder is also very focused on reading other people's faces. So if I have some social anxiety right now, but if I was really socially anxious, I would pay attention to quite a lot of your faces right now to try to find confirmation that you look at me and think that I'm actually doing something really bad, or that I look incompetent and those things. Okay, so that was a very simple design, simple experiment, and the heritability for variability outperforms the standard mean bold. Of course, here's a range. So what you see here is actually the heritability ranging from 0 to 1, and the peak maximum is 0.65. So of course you would find not the average is 0.25, but also some peak regions, as the fusiform gyrus occipital lobe being the most heritable for this type of experiment. We have tried to do this with trying to predict treatment response. So there is this idea that maybe it's not possible for us to know to find one treatment that fits all. Maybe that's will never be the case. But what if we could predict who will actually respond to treatment even before providing them with treatment? So when I seek treatment at the healthcare, I want to know what is the likelihood of me responding to this particular treatment, not because people in general seem to respond. And I think this, if we could end up there having individualized and personalized predictors, that would be of course very helpful for clinical decision making. So we took our estimation of neural variability at baseline, and we did this with again with the social anxiety disordered patients. We scanned them twice before they even had any treatment. So it's just an 11-week waiting period between the first and the second scan. And our idea was to find reliable activity before treatment that could later be predicting their response to treatment. And we did this with both task-based fMRI and resting state fMRI. What we found was that our task-based neural variability outperformed the resting state. So even even a very short and very simple task, in this case showing emotional phases, could do a better job than our resting state. Comparing that to again same data, but the good old conventional estimation of mean bold, which didn't contribute at all significantly to the prediction model. And at last, in the red bars is the LSAS, which is the self-reported measures. So even, I mean, how people self-report their anxiety level at baseline was also contributing to that the treatment outcome. But we could, resting state was able to do the job, but then we had to add much more data. So what you see here on the x-axis is actually time, or volumes. So in this case, 340 seconds of data, whereas we only needed 160 for task. But we even did more of that, so we trimmed the data even down to 40 seconds of data, and our task-based fMRI remained both reliable and able to significantly predict that treatment outcome. If we added several, so having task and resting states, and also self-reports, the prediction model also improved the accuracy. So lower value means better accuracy. And again, this idea of maybe we don't need to add more data, maybe we can have just less data and the model can do the same job. And this is the same graph that I just showed you on prediction accuracy. And if we look at the spatial pattern for what regions that contributed to that outcome, that spatial activity, activity across the whole brain remained stable, even for a 40-second scan. So maybe we don't, I really want to push the field in trying to use better metrics and maybe even shorter time, because that may be more feasible for us with certain patients. And this is another graph also showing the test-retest reliability of our prediction model. So remember in the beginning, I showed you two meta-analyses of test-retest reliability within the fMRI field, one on functional connectivity and the other one was on task-based fMRI. And those models are down here to 0.3, 0.4, whereas our prediction model improved. So the more time you include, the better accuracy, especially for the resting state model. But our task data remained doing a good job even after 80 or even 160 seconds. I think it's my last slide. I want to give you another example of, I criticized myself in the beginning saying pre and post designs are not, it's not enough. So what I try to do here is to scan our patients multiple times. So in this case, I actually had patients in the scanner 5 sessions. So I had them to come back twice before they had the treatment. So this is the test-retest period that I talked about before. And what I show you here in the graph, this is self-reported symptoms of social anxiety. So of course, the lower you score, the better the less social anxiety you have. So between the two test-to-test periods, there is nothing happening. It's just an 11 weeks of waiting for treatment to start, which initiates here. And then we have them to score their anxiety symptoms weekly. And this weekly when they are having cognitive behavioral therapy. As you can see in the early phase of the treatment, the four first weeks of treatment, symptoms doesn't drop much at all, but significantly symptoms drop in the later phase of treatment, which is also expected because that's when we do certain types of components in the treatment, which is called exposure-based interventions. So symptoms change later on in therapy, and symptoms remain stable in the early phase. So since we did imaging twice before treatment, we did imaging in the halfway through treatment, and then at post-treatment. And actually, we also I had them to come back for a 2-year follow-up later, but I'm not including that story here. So when we look at changes that could happen in the brain throughout all these time periods, well, the test-retest period was stable, so nothing really happened. But everything that could happen across the early and late phase, the brain's neural variability changed the first 4 weeks, and basically nothing happened after that. So in the later phase, so the brain responds and changes early on, and is also able to predict the clinical score. I don't include that plot here, but the early change in variability predicts the later change in symptoms. So it's kind of like the brain is one step ahead, and you could say, well, maybe 4 weeks of intervention was enough, but based on all our clinical experience and all the clinical trials with this type of treatment, we know that the treatment would not work if we stopped halfway through. We need to continue doing the rest of the treatment as well. But again, there is something in the brain that prepares us and makes it possible for us to respond. And some of these responses, especially in the medial prefrontal cortex, was also different between our patients and a healthy controls, group of healthy controls. And after treatment, these differences at baseline was not apparent anymore. I'm just gonna finish off because I know the time, and I'm happy to answer some questions. Okay, so summary, task-based neural variability. I showed some examples how this is reliable. It's less sensitive to data volumes. We may be able to acquire less data, get the same result. It differs between mono and dizygotic twins. It may be able to predict outcome, and it seems to change during treatment. We have some work in progress, ongoing data collections. Of course, we need volumes in terms of patients. So we're right now in one large trial called TREVAR, completed more than 160 patients at pre-treatment. And we also recently got funding to do a similar approach with adolescents in Romania. So we'll study adolescents with anxiety and depressive disorders. Next steps for me and my group is to also test this with another disorder, namely major depressive disorder, when they had ketamine or placebo treatment or SSRI treatment. And another trial we also recently got funded from Vetenskapsrådet. We will study the placebo responsiveness and neural variability. So I think that's this. Yeah, thanks to my team and people that helped out. And thank you for your attention, and happy to answer.
Thank you for that, Christopher.
Yeah, so my question, I'm not sure if I understood correctly what you said, but so you mean that with your scanner at 4 weeks after treatment onset, basically you would be able to predict whether the treatment is going to work or not for this specific patient?
Well, for the prediction, that we did even at pre-treatment. So the prediction of outcome, that we did even before the treatment started.
Even before the treatment started, you know if it's going to work or not?
Well, not perfectly, but
you have a good indicator.
Right, but when we then looked into how brain changes, we basically saw all changes happening in the first 4 weeks.
And so you said you were not going to talk about the 2 years follow-up. Is it because you didn't have time to look at the data, or just for time's sake now?
For time's sake, but I can. It stays. It aligns very well with what we found here.
Okay, cool. Thank you.
I'm sorry, you
Oh, well, I was wondering, in this intervention, did you use a cognitive behavioral
Cognitive
therapy
behavioral
only?
therapy is the data that I presented.
With or without combination with SSRI?
Without, yeah, without. So there, it's a very clean sample of patients who had no other intervention. Actually, 4 individuals had SSRIs for years before starting the intervention, but they had no changes in the dose or anything throughout the whole trial. But that's 4 out of 50.
Is it established that I have read the best results with the combination of both psychotherapy and pharmacological treatment? Have you tested this or compared with?
Yeah, but I only have 4 individuals, so
Okay.
it's not possible. And it's also depending on what the combining the two may be good for some patients. Others, there's no real benefit.
So these predictive results that you report, is that based on the variability of response across the whole brain? And if you were to kind of preliminarily try to zoom in and see if there are certain regions that are more informative than others?
Yeah, thank you for a very good question. I didn't stop, but I mean, really just touched this topic. Yes, very importantly, whole brain. So I make no restrictions to whatever region or nothing. I just look at everything that could be there. But I have one criterion, and that's the reliability. I want the activity to be reliable across 11 weeks. That was what's one. And the most that region that contributed the most to the prediction of outcome, that was the primary V1 occipital and occipital cortex. And that's what we're trying to dig even deeper now into, understand what that may be. We speculate about that this is what's been known but what we thought about how your visual cortex response and may also be a bias in how you interpret or how you perceive information from others. And in this case, it's emotional phases. So we did talk a little bit about that in that paper, but now we're also collecting a lot more new data to see if we can.
So on the face of it, that's kind of surprising. Like you see in the primary sensory area, you see a response to cognitive therapy.
Yeah, not response to treatment, but there seems to be a process that may be related to why you respond.
Where the exclusion criteria like autism, personality disorders, for example, psychopathy, they used to have hypoactive amygdalas. Those patients,
right?
Probably they don't have that much social anxiety.
Right. So no, it's in the data I presented here was a very clean social anxiety disorder samples. They had less comorbidity with other personality disorders, so but depression was the main comorbid.
Oh, yeah, yes.
So I just have a question. So this work falls into the debate, as far as I'm understanding, about whether we need a lot of data or not, basically, to get useful results. But honestly, I'm a bit not sure what is the point of this debate, in the sense that I don't think there's. I mean, I would think, but I might be wrong, that there is no one approach better than the other. They just give different answers, and also depending on which type of choice you go for, then as long as you correctly apply, you correctly interpret the result, make the right research question, and also they involve different statistical approaches, and both have pros and cons, of course. So I don't see really the point of this kind of debate about whether it's needed to have more data or less. I don't think there's a need. It just depends on the question you want to answer, right? Or
right.
am I missing something new.
So first I want to stress that we need more data in terms of individuals. We need more individuals to make accurate prediction that actually can generalize to new patients. I mean, we've done that now with using different cross-validation techniques, and but still we have not been able to test that with training and training a model that applies to a completely separate sample. So, and in order to do that, you need samples. So there's no question about that. The other is that, well, people have really been arguing a lot. Well, the reason why we have so low test-retest reliability in general is that we have sampled too little, and we just need to scan patients for longer times. You would see that come up every now and then, and people are pushing that in that direction. And of course, I am having a hard time convincing the clinics that I work with now to include imaging in their clinical practice in order to recruit patients that we can scan. And I would not succeed if I'm going to say, I need them for 3 hours in the scanner. So it's not going to happen.
All right, sorry, one final.
Like very quick questions about the prediction model. This might predict the effectiveness of the therapy. So what kind of therapy is it? Is it like drugs or?
Yeah, so the data I presented was cognitive behavioral therapy. And one main component here is exposure-based interventions. So for socially anxious individuals, it's actually to prepare them in order to do a thing like I'm doing now, giving a talk in front of others. So you expose them to something that they've been avoiding for many years. And actually to just stay in that feeling of being anxious and in order for you to experience that this anxiety and all the bodily symptoms and all the cognitions and everything that's happening will actually taper off naturally just by staying in this situation. That is the main component.
All right, okay, fine. Okay.
I was just wondering about what you commented on the visual cortex. So do you think if you had, let's say, an auditory paradigm, you would see auditory primary cortex pop up? So since you look at variability, could it be that those are regions that need to respond flexibly? So, you're biased towards variability, whereas if you had looked at the mean, maybe, amygdala would have been important because it's just tonically active. And maybe that explains why your combined score was even more effective, because then you can pull down, variability from V1 and tonic from amygdala, etc.
That's an interesting point. Yeah, so that is, I actually ended up not including auditory stimuli in the new trial, but we were really considering that. We do now include much more different types of stimuli. So I didn't focus much on that here now, but not only emotional information, but non-emotional processing and higher and lower level of perception. So hopefully I can get and have a better answer in the near future. The other point you make is also very interesting about the tonic response of amygdala, because that amygdala had nothing, the variability, we found nothing contributing from the amygdala. But when we do, yes, exactly like you say, when you do mean bold comparing emotional to non-emotional stimuli, we typically see amygdala response, but not for variability. Yeah. So actually one of the things that I'm also been trying to understand how that could be, but thank you for a good question.
Thank you.
Thank you.